So now I will be starting with viruses. Term virus, it was coined by the scientist Pasteur. Now, for the first time, discovery of virus was performed by the scientist Ivanovsky. The virus which he discovered, that is TMV, which is tobacco mosaic virus. First time discovery of virus was done by scientist Ivanovsky. Virus which he discovered, that is TMV, that is tobacco mosaic virus. Yes, TMV. Now, tobacco mosaic virus, it was later crystallized by the scientist Stanley. Yes, so you write name of another scientist, that is Stanley. He first time crystallized tobacco mosaic virus. Now another scientist is Bacteriophage. Bacteriophage, he called these viruses as contagium vivum fluidum, which means living infectious fluid. Bacteriophage called virus as contagium vivum fluidum. Yes. What that means? This means living infectious fluid. Living infectious fluid. As you know, viruses, they are at borderline of living and non-living. So now I will be dealing with certain features of viruses which are living or either non-living. Features of viruses which resemble non-living entities. Like the living organism in the virus, protoplasm is absent. Protoplasmic constituents, cell organelles like chloroplast, mitochondria, all these organelles, they are absent. So there is lack of protoplasm. In the virus's respiratory system, which is also known as the Lipman system, for the synthesis of ATP is absent. Respiratory system involved in synthesis of energy currency of cell, that is ATP, is absent. Viruses, they can multiply, but they can multiply only inside the body of host cell. They can show multiplication only inside the body of host cell. Independent existence of viruses absent. Another feature, independent existence of viruses absent. So you can call them as obligate intracellular parasites. They are obligate intracellular parasites. Ability of growth, that is increase in the size. Increase in the size for viruses is absent. They do not have ability to grow. Like non-living entities, specific gravity of viruses is high. Specific gravity like non-living entities is high. Now, like the non-living entities, the viruses, they can also be crystallized. Just now I have discussed, for the first time, virus was crystallized by the scientist Stanley. They can be crystallized. So these all are the features of viruses which resemble non-living entities. Now let's discuss feature of viruses which resembles living beings. Viruses, they are made up of organic compounds. Organic compounds. Viruses, they contain genetic material. Either it can be DNA or it can be RNA. Presence of genetic material. In the viruses, organic compounds, they are present. Organic compounds which are present, one amongst that is genetic material. Along with that, there is also presence of proteins. And some of the viruses, they are associated with catalytic proteins. And you know these catalytic proteins, they are enzymes. Enzymes which can be associated with the virus, they are lysozyme. Yes. One of enzyme I am writing here, that is lysozyme. Lysozyme we have studied in the previous class with respect to transduction in bacteria. Another one I am writing here, that is reverse transcriptase. Now viruses, they are pathogenic, they are infectious like living organisms. Like other living organisms, they are infectious. They are pathogenic, disease causing. Viruses, they have ability to show changes. They have abilities to show mutations. Like the other living organism, the viruses, they can also be killed. They can be killed by autoclaving. Autoclaving is high temperature treatment and with the help of UV rays. Now after this, now I will be dealing different components present in the viruses in little bit detail. In the viruses, maximally there can be presence of 4 components. One, genetic material, that is nucleic acid. Now this nucleic acid, it is covered, it is protected with the help of proteinaceous covering. Nucleic acid is covered, it is protected with the help of proteinaceous covering. This proteinaceous covering is known as capsid. In some of the viruses around capsid, there can also be presence of envelope. In some of the viruses, one more covering can be present, that is envelope. And as I have discussed right now, that in the viruses, there can also be presence of catalytic protein. Catalytic protein, that is enzymes. So I will be starting here with the first component, that is nucleic acid. Now nucleic acid can be DNA, or RNA. Either DNA or RNA, nucleic acid, is present in the viruses. If virus contain DNA as nucleic acid, then such type of viruses, they are known as deoxyviruses. You got this? अगर वायरस के अंदर DNA present होगा as a genetic material, so such type of viruses, they are known as deoxy1. But if genetic material is RNA, now you will be calling them as riboviruses. Now DNA which is present in the virus, this DNA can be DS that is double-stranded, or this DNA can be SS, that is single-stranded. So I will be writing here example of few viruses which contain double-stranded DNA. One example for this is pox virus. Another one, CAMV. CAMV, please write full form of CAMV. That is cauliflower mosaic virus. Cauliflower mosaic virus. Now ssDNA, that is single-stranded DNA. This is present in virus, that is 5X174. Another example, M13. Now coming to riboviruses. In the riboviruses, again, RNA can be SS, that is single-stranded, or it can be double-stranded. Single-stranded RNA is present in virus which was first discovered, that is TMV, tobacco mosaic virus. Along with that, also present in influenza virus, foot and mouth disease virus, and HIV virus. HIV, that is human immunodeficiency virus. This is AIDS virus. So please write full form, human immunodeficiency virus. Now this HIV virus, this comes under category of retroviruses. Yes, please note this down. Please note this. HIV virus come under category of retroviruses. Now what do you mean by retro here? I have discussed in the previous classes that DNA contain information. It contains coded information for the synthesis of RNA. DNA से RNA synthesize होता है. And what this process is known as? This is known as transcription. But in the retroviruses, genetic material which is present, that is RNA. The genetic material which is present, that is RNA. In these viruses, RNA can be converted into DNA inside the host cell. Yes, from the RNA, DNA can be synthesized inside the host cell. So, ये जो upper process है, इसको हमने कहा transcription. So, surely now conversion of RNA into DNA, this is known as reverse transcription. They show reverse process. They show retro process. That's why they are known as retroviruses. To perform this process, they have enzyme, that is reverse transcriptase. This process was first discovered by scientists Temin and Baltimore. Temin and Baltimore. That's why this reverse transcription It is also known as terminism. So the concept of retroviruses is clear to all of you. Retro means reverse. They are showing reverse transcription, conversion of RNA into DNA. Now after this, now some of RNA viruses, they can also have double-stranded condition. So you write one name here. Few examples here. One is reovirus. Reo, they attack respiratory system. Another is wound, tumor, virus. Now, viruses which attack plant. Yes, viruses which attack plant. Generally, these viruses, they contain single-stranded RNA. I'm writing this information here. Viruses which attack plant. These viruses, they are known as phytophaginae. They generally have single-stranded RNA. Now, viruses which attack animals, they are known as zoophaginae. Zoophaginae, and in the zoophaginae, there can be presence of double-stranded DNA, single-stranded or double-stranded RNA. Viruses which attack bacteria. Bacteria, they are bacteriophages. Bacteriophages. Now, in the bacteriophages, the type of genetic material which is present, that is dsDNA, mainly double-stranded DNA. Now, after looking into this part of virus, that is nucleic acid, now I will be dealing with protective covering around it, that is capsid. Now, capsid is proteinaceous in nature. Capsid is made up of subunits. Capsid is made up of proteinaceous subunit. These proteinaceous subunits, they are known as capsomeres. Right. Number of capsomeres associated with capsid of each virus, they are fixed. Number of capsomere associated with each virus, they are fixed. I am citing one example here. For TMV, in the TMV, number of capsomeres which are present, that is 2130. Now, arrangement of capsomeres, they provide shape to the virus. Yes, arrangement of capsomeres, they give definite shape to the virus. Shape of the virus is dependent upon arrangement of capsomere. So here I will be discussing shape of TMV and bacteriophage. Let's start with the TMV. In the TMV, there is presence of elongated shape, cylindrical shape, like this. Elongated, or you can say cylindrical shape. ये जो मैंने कवरिंग यहाँ पर फॉर्म करी है, this covering is capsid made up of subunits. Yes, covering is made up of subunits. So these subunits which I am making here, these are capsomeres. So you're getting that? ये जो मैंने यहाँ पर सब यूनिट्स बनाई हैं, which are forming capsid. These subunits, they are capsomere, and number of capsomere associated with TMV is 2130. Capsid के अंदर there is presence of RNA, genetic material. So I am showing here some part of RNA. Yes, single-stranded RNA. Now next one, that is bacteriophage. The structure of bacteriophage, it is known as lollipop-like structure. The structure consists of head and tail, so also known as tadpole-like structure. Now, in this structure, there is presence of head, तो ये जो पार्ट मैंने यहाँ पर फॉर्म करा है, दिस पार्ट ऑफ वायरस इज हेड. इट इज प्रोटीनेशियस इन नेचर, सो यू कैन सी द शेप ऑफ हेड. द शेप ऑफ हेड इज पॉलीहेड्रल. इनसाइड हेड दिस प्रेजेंस ऑफ जेनेटिक मटेरियल. हेड के अंदर प्रेजेंट है जेनेटिक मटेरियल, and genetic material which is present here, that is dsDNA, double-stranded DNA. Along with the head, there is also presence of tail. Yes, head के साथ there is also presence of tail. You can see tail is elongated. Tail is elongated. Head and tail, they are joined with the help of collar. They are joined with the help of collar. Yes. Along with that, one more structure. This one is neck. Collar and neck, they will be joining head and tail. Around tail, there is presence of contractile covering, contractile sheath. So, around tail, I am forming this sheath. This sheath, it is contractile. Contractile sheath. At this end of tail, there is presence of hexagonal basal plate. तो यहाँ पर हम बनाएंगे plate which is hexagonal. This is known as basal plate. Basal plate, you can see 6 edges. 6 edges are there. ये जो basal plate है, इसकी 6 edges के ऊपर present है tail fibers. तो ये जो मैंने structure बनाए, these are tail fibers. And नीचे वाली side पर, on the lower side here, on the lower side of plate, on the lower side of edges, there is presence of these structures. These structures, they represent pins, tail pins. So total, you can say 6 tail fibers and 6 tail pins. They are associated with the structure of bacteriophage. तो जैसे अभी हमने यहाँ पर पढ़ा, tobacco mosaic virus shape is elongated, but in the case of bacteriophage, some part is polyhedral in shape, some part is elongated in shape. Such type of virus which is associated with 2 types of shapes, it is known as binary type of virus. This type of condition, it is known as binal type. Now after looking into the capsid, now another part which I will be dealing here, that is envelope. So now I will be dealing with next component, that is envelope. Envelope, it is made up of lipids, carbohydrate and proteins. Among these three components, lipid and carbohydrates, they are contributed by host cell, and proteins, they are coded by viruses. Now the viruses where envelope is present, जिन viruses के अंदर envelope present है, so these viruses, they are known as enveloped viruses. One example for this is HIV. Viruses in which envelope is absent, these viruses, they are known as naked viruses. One example for this is poliovirus, another one, reovirus. Like capsid, this envelope is also made up of subunit, and these subunits, they are known as papillomeres. The subunits of envelope, they are known as papillomeres. So here I will be making diagram of HIV virus to show envelope. In the HIV virus, there is presence of single-stranded RNA. 2 copies are there. Single-stranded RNA की 2 copies है. Around this single-stranded RNA, there is presence of capsid. तो इस RNA के आसपास आप बनाएं capsid. Around the capsid, now there is presence of envelope. Yes, around the capsid, one more covering. That is envelope. Yes, this one. Now this is outermost covering of HIV virus, that is representing envelope. Envelope can be smooth or it can have certain outgrowths. तो यहाँ पर जो HIV का envelope है, यहाँ पर ये आपको जो structures नजर आ रहे हैं, these structures, they are known as spikes. Now, after looking into the components of viruses, now I will be dealing reproduction in virus. Reproduction of virus is of 2 types. That is phagic and phenocytic. In the phagic reproduction, only genetic material of virus enter inside host body. Only genetic material of virus enters host body, enters host cell. But in the case of phenocytic type of reproduction, there is entry of complete virus inside the host cell except envelope. Both genetic material as well as capsid enters host cell. But envelope will remain outside. For this, I can write one example, that is HIV, which show this type of condition. Now, phagic type of reproduction is of two types, lytic and lysogenic. I have already discussed these two types of reproduction while dealing with transduction. In the lytic type of reproduction, you know, in the lytic type of cycle, virus kills, virus break bacterial cell. It causes lysis of bacterial cell. तो यहाँ पर सेल का lysis होगा, host cell का. Let's say example of the host cell is bacteria. Viruses which can break host cell, they are known as virulent viruses. One example for this is bacteriophage, that is T2. In the lysogenic, let's see what will happen. This is, let's say, bacterial cell. ये है बैक्टीरिया का डीएनए और वायरस का डीएनए बैक्टीरिया के डीएनए के अंदर इंटीग्रेट कर गया है. Virus DNA has shown integration around in the bacterial DNA, and this condition is known as prophage. Now, जब तक वायरस का डीएनए बैक्टीरियल डीएनए के अंदर इंटीग्रेट रहेगा, this virus cannot do any harm to bacteria, any harm to host cell. This type of the viruses, now they are known as temperate viruses. And one example for this is lambdaphage. Now, after looking into the reproduction, now I will be dealing with the entities which are lower than the level of virus. Some of the entities are there which are lower than the level of virus, that is viroids and prions. So now I will be dealing with subviral agents. That is the entities, agents which have size lower than viruses. One example for this is viroids, and another one I will be dealing here, that is prions. Viroids, first time discovered by Diener in 1971. Prions, they were discovered by the scientist Alper. Now, viroids, they are infectious RNA particles. Yes. Now, the capsid protective covering is absent. They represent infectious RNA particles. Infectious RNA entities. RNA which is present, that is low molecular weight and single-stranded. But in the case of prions, they represent infectious protein particles. They are infectious protein particles. Now, genetic material, RNA or DNA, both are absent. Viroids, they are infectious, but they can cause diseases only in plants. They are pathogenic to plants only. Plants, prions, they can cause diseases, but only to the animals. So I will be writing certain examples for diseases caused by the viroids in plant. One disease is PSTD. That is potato spindle tuber disease. Please note this down. Potato spindle tuber disease. Next one, citrus exococcus. Another one, cucumber, pale yellow. Then chrysanthemum, stunt. Now diseases caused by prions. One is Kuru disease in humans, that is also known as laughing death. Another one, scrapie. Scrapie's disease in sheep. All these diseases, they are associated with nervous disorder. Another one, bovine bovine spongiform. Encephalopathy, that is BSE. This is also known as mad cow disease. Please write this, mad cow disease. Next one, that is Creutz's. Felt, Jacob disease. Jacob disease, that is CFJ. So all these diseases, they are caused by proteinaceous infectious particle. So this is all about viruses as well as subviral agents. So in this class, we studied mycorrhiza, lichens, and last viruses and subviral agent. This is all about biological classification. In the next class, I will be starting with the new chapter. Thank you so much.